This is a very generality of what it is, but I think even vertebrate immunologists sort of recognize some things like phagocytosis and production of reactive intermediates of oxygen and nitrogen and antimicrobial. I prefer to call them immune peptides and antimicrobial, but they do a lot of things that vertebrates do, and they're pretty effective in protecting them from foreign insult, okay? The thing that we've spent most of our life doing is this response called melanization. You know what melanin is. It's that brown pigment, right? Okay? And you can actually get melanomas or cancers because these things go crazy. Well, in insects, they use melanin for lots of different things, and they share this chemistry. They tan their cuticle, they harden their eggs, they heal their wounds, and I think they've adapted this chemistry for defense. Then they use it to kill parasites. At least some of them do. One of the mosquitoes that does it very well is an Asian mosquito that we affectionately call Armigeria subalbatus. And the Brugia filarial worms, I mentioned Brugia malayi, which is a human pathogen. Well, Brugia pahangi is kind of a cat parasite. Doesn't affect humans, although there's stories of old where they could actually inject some into some natives at a time when they didn't have IRBs and determine that it could actually become infected, okay? But this is a nice model because this mosquito actually kills all Brugia malayi that ingest by melanotic encapsulation, and it's permissive to the development of Brugia pahangi. The Japanese used to use this mosquito to separate the species of parasite if they didn't know which they had, and it worked very well. So here we have melanization functioning as a natural response, and you can see how this system would be a pretty nice one to study. And so our laboratory mouse is really the mosquito. It's just a little smaller. But we can put things in and take things out and do all of these things, and that's why I show you this picture. You can manipulate it. You just have to practice manipulating it. Okay? So here's a couple of little slides. On the left, you see this worm. I don't think I have a pointer here, do I? Is there a pointer? No. Anyway, up in the left, you see that worm with the cells around it. That big thing down there is called salivary glands. If you look at a closer thing of that worm, you can see it on the right side. That's where you dissect one out. So you have this, hemocytes are like the white blood cells of a mosquito, and they're wrapped around this thing, and they've laid down this layer of melanin together with structural proteins to sort of entomb this parasite. And if you look at it ultrastructurally, this MF is the parasite, and then you see this dark pigment on it. That's a great big granular cell, okay? Now, Mike Strand knows lots and lots about insect cells, blood cells. So if you want to get details on what these things do, ask him, don't ask me. On the right side then is a fully melanized transmission electron micrograph of a parasite that's dead, and this thing is entombed in all of this cellular debris, and then it gets walled off and it does its job. It became very popular when a science article came out with some work they did at the NIH where they selected a strain of Anopheles that was totally resistant to malaria parasites, lots of different malaria parasites, because it would melanize the parasite on the midgut of the mosquito. It took a while for people to actually realize later that the only plasmodium it didn't melanize was the plasmodium from Africa, which is, of course, the only place where this mosquito lives. So, again, coevolution is pretty good. So, most of my life, I spent sort of figuring some of this stuff out. So we've, done a lot of biochemistry, silenced a lot of genes, cloned a lot of stuff, and determined the importance of all of these things. But, so what? So we ask the question, I think, that all of us sometimes ask in our life. I know Dan and I have talked about this. It's like, so what? Does any of this mean anything in the real world? Is anything that I do actually going to have any impact on how these pathogens actually affect people in the real world? So, now we're going to get to Papua New Guinea. So actually they had a situation there that there was a vector biologist there by the name of Moses Bukhari, who since left. He's now at Liverpool, and he is sort of the voice for the global program to eliminate lymphatic filariasis. So he took over a position from another guy that was as equally fanatic as Peter Hotez. So, but he was in Papua New Guinea at that time, and he asked my good buddy C.C. Chen from Taiwan and I if we'd come over and look at some of their mosquitoes in terms of vector competence and whether or not there was some way that these mosquitoes were actually killing the parasite, because he thought they should have more parasites in the mosquitoes than what they actually did. It didn't seem right. So anyway, we went to Papua New Guinea, and I'm not questioning any of you people knowing where Papua New Guinea is, but the first time I went many years ago, I called my bank to see if I could get any kinas before I went, which is the denomination, and the woman in the international market told me she was sorry, but they didn't have any African money that she could give me. So it's not in Africa. It's quite close. You notice there's another half of New Guinea, Indonesia and New Guinea, and nobody goes in there and does anything. So I'm not sure how much you do in Papua New Guinea is really going to matter in the long run. It's a fabulously interesting country with a tremendous diversity in geography and ecology, much of it because it's very close to the equator, and then it just rises out of the ocean up to about 4,500 meters. So you have very cold, high mountain areas, and then you have very, very warm, low areas. You have a population of about 6.5 million people, but yet it has about 15% of the world's languages. So if you think about strategizing for a public health program, think about a place where you can't get any place because there's no roads, and then everybody speaks a different language. It's pretty interesting. They do have a uniform language called Pidgin. The health statistics are awful. Think of a mortality of children having 30,000 of a population of 6.2 million dying before they reach the age of 5. That's pretty catastrophic. So if you're born in Australia, you're going to live 25 years longer than if you're born in Papua New Guinea. So pneumonia and malaria are bad, but HIV now is becoming more of a problem. Filariasis is bad. They were kind of smart, I think. In 1968, they established what's called the Papua New Guinea Institute of Medical Research. And this is a program that actually says if we have limited resources, shouldn't we at least find out how we should use our resources? And I think the strategy is very good, and it was really good because now most of the money coming into the Institute of Medical Research actually comes from places like the NIH and the Wellcome Fund, and so it's all of the foreign scientists that go in there and work that bring the money and actually support this at a level much higher than what it would be anyplace else. So about 1 million of the people are infected with filariasis, so you have about a 20% prevalence, and most of the transmission is in the lower area. Malaria is endemic in all 20 Papua New Guinea provinces, and you know, you learn in parasitology class that you rarely, if ever, see Plasmodium ovale. You know, well, there's some areas in Papua New Guinea where 6% of the population has ovale, and what I'll show you later, you'll find out is that many people have several different parasites at the same time. So the principal vectors of these parasites, of both malaria and lymphatic filariasis, are mosquitoes in the Anopheles genus, and most of them in what we call the Anopheles punctulatus group. So there's Anopheles punctulatus, Colianensis, and a number of species, maybe more than 6 now, of Fawadi. And I'm not going to require you to learn all of these, but just understand that to separate some of these species, you actually have to use molecular techniques to be able to do it. And each one of these has a distinct and different biology. And for example, here you can see Anopheles punctulatus collections on the left. And when we're out in here, this is in the open sunlight. That water is about 95 to 100 degrees Fahrenheit, and the mosquitoes are happy, okay, the larvae in there. If you go to Fawadi 1, you'll find that along the coast because it likes shaded areas and brackish water. It likes a little bit of salt in the water. So very, very different biologies, very different habits and habitats. And I think we're going to find out that they transmit in very different ways probably and have different susceptibilities. So in the transmission dynamics, there's a concept of facilitation, which basically says if you have more microfilariae circulating in your blood, you'll get more parasites into mosquitoes. And that seems pretty simple. But it doesn't always work that way, and it wasn't working that way in Papua New Guinea. There was limitation. As you got to higher numbers, you actually got fewer parasites, and nobody knew what was going on. There were some theories, but so we went to play. Here's some of the transmission dynamics that they knew, and we'll kind of gloss over this. Just realize that in some of the villages, like everybody is infected with Wuchereria bancrofti. I mean, literally everybody is infected. The only reason we didn't know that before is we didn't have a sensitive enough molecular diagnostic technique. In some areas, the transmission is potential, even with a bad vector, that people are being exposed to over 2,000 larvae in a single year, which is pretty amazing that not more people are infected. You should have higher transmission dynamics than what you have with the number of parasites you have circulating. So we went in to do some work, and I'll tell you what we did first, and then I'll tell you where it's led us. So the only way you get into Papua New Guinea is fly into Port Moresby, and you can only fly into Port Moresby by Air Nia Guinea. If you're going to go anyplace else from there, then you have to fly, because what I'm showing you here in yellow is the highway system in Papua New Guinea. So you can't. So we went to work in the way upper right. Wewak is, we flew into Wewak, and then we went to a field station that the PNG IMR has in a little community called Maprik, okay? And then much of the work we do since then has been in a facility in Madang, which is actually a laboratory facility. And up that north coast, as you go towards Awar, is the little village of Bogia, where it's never been tested before. You know, Kazura has been there since 1978, and we find villages right along the road that nobody has ever tested for filariasis, and there's a prevalence of 60% and all kinds of big legs running around. So, but the roads aren't necessarily always good, okay? They're a little better in the dry season than they are in the rainy season, but the scenery is spectacular. So driving on bad roads is made up for the fact that you see primary tropical rainforest, which is, for any biologist, is an absolute treat. This is the field station in Maprik, and it's pretty nice. The housing is pretty nice, and it's free if you go to work. You have to cook your own food. And fortunately, this is my good friend C.C. Chen, and they have a wok there, and as long as there's a wok, he can cook anything, okay? So he cooks, I clean, and I buy the beer, and it works out well, okay? On the way to the village, we stop at a typical store. This is about what you get for a store, which is a little roadside place, and it's got bananas, but what we're actually buying is a betel nut. You see those on the lower thing. Betel nut here is from the betel nut palm, and it contains a psychoactive alkaloid, so it's a drug that they chew. They chew it throughout Southeast Asia. In Taiwan, they chew it a lot. Vietnam, they chew it a lot. Thailand, Laos. It's a very popular drug. You can tell, even you tell these little girls are chewing it that I took the picture of, because they have the powder on their face. The powder is actually a lime that they dip it in. And the mineral lime, if they dip it in, it increases the psychoactive activity of the thing. And then you can also tell when people chew it, because over time their teeth turn red. So it stains the teeth red. So the government is trying to keep people from chewing. We buy betel nuts when we go to a village, because there's no better impression you can make with the people than to give them betel nuts, you know, because then they're really happy. So the tree that had the betel nuts there, remember, that's the field station that we have. So the betel nut palm is growing around outside. So this is a Drekakir village complex, probably the complex that has been studied probably more than anything else. It's where they actually eliminated lymphatic filariasis, and I think it ended in the around late 90s, I think. It was the first one ever done that Jim Kazura did. Well, we went back there in 2006, I guess it was, or 7, and that's where we collected all our infected mosquitoes, so they have as much lymphatic filariasis as what they did before. A very happy people, but you can point out some potbelly children, so obviously soil-transmitted helminths are pretty abundant there, as they are throughout most of the tropics. I had dinner one time with the Prime Minister's daughter, who is sort of the public policy sort of advertising for her dad. And she told me an interesting thing at lunch, in that she said Papua New Guinea the economy runs on 3 things. It runs on land, land, women, and pigs. So I have this picture of the 3 things. So villages oftentimes barter all of those things. So land is very precious to them. Pigs are very precious to them. And so a lot of diseases associated with pigs are also lots of neuronal cysticercosis, for instance. And then women are oftentimes exchanged between villages so that they can create better feelings, I guess.